So this talk is about algorithm for computing something called the gene tree probability, and the model is so-called multi-species coalescent. Let me first give some introduction about the coalescent, which is one of the very important concepts in population genetics. This coalescent you can see from this one of the well-known model in population genetics called the Wright-Fisher model. And in Wright-Fisher model, we have a number of alleles. So it's like a gene copies. And these alleles are arranged in generations. So, the time flow from top to down. And so each row is a generation. And the basic assumption in Wright-Fisher model is the generations are non-overlapping. And the population size do not change. It's constant population size. And it also assumes the sort of random mating. So in other words, if you see this generation after generation, and when each generation dies, and then you create a new generation, and every generation the alleles is taken randomly from one of the allele in the previous generation. So this is called random mating, and the coalescent you can see by considering the history of the samples. So suppose you sample these three alleles, and you can only afford to study a small subset of samples. So let's say you consider this what happens to these three sampled alleles, and then this is in the current time, and you trace backwards, and you can see where they come from, and they can trace from previous generation. You can continue to trace. Then once you're here, something interesting happens. You see these two find the common ancestor, and this is called a coalescent. And you can continue to trace, and then on here, and all three samples find the common ancestor, and then you can continue. So this is the basic concept of coalescent. And the importance of a coalescent is that this is a stochastic process, pretty much determining how you're going to observe the gene genealogy. That is how these sampled alleles are related to each other. So in this case, the two alleles on the left, they are more closely related. So let me tell you what is a coalescent in the sort of multi-species coalescent, or coalescent in the multiple populations. So here it shows three populations, A, B, C on the top. And these big, very thick trees are usually called species tree, or I sometimes also refers to as a population tree. So they really refers to how these populations are related to each other, like when they diverge and how closely they're related. And then let's say, what does a coalescent do? The coalescent will actually determine if you consider samples from the populations, and how this sampled alleles within populations is determined by the coalescent. And in particular, the multi-species coalescent, in every current or ancestral population, you actually run a separate coalescent process. So you see from here, suppose you sample from these three alleles, the darkened allele, and you trace backwards in time. Now it's time flow the other way, and then you can continue. And now you see these two coalescent here. So this is how the coalescent will determine how this genealogy of a sample allele will be like when you have multiple populations or species. And you can continue. This is coalescent, then continue, and continue. So there's a well-known phenomenon. It's called gene tree and species discordance, which means the gene genealogy may not be the same as a species tree or the population tree. And one of the reasons may just because this is a stochastic process, which is underneath this multi-species coalescent. And in fact, if you see here, whether you observe this coalescent of a B and C lineage from B and C really determine how long the time T is. If you have lots of time to coalesce, then this coalescent is more likely to occur. Otherwise, it may not. So it's a lots of stochasticity here. So this is the event. So the multi-species coalescent allows computation of the probability that we observe specific gene genealogy based on the coalescent theory. All right. So let me define what is the main subject of this talk: the gene tree probability. The gene tree probability is that we're given a species tree, so we're given the species tree with a branch length and also topology. And we're also giving a gene tree. So this is a gene genealogy that doesn't have a branch length, only have topology. It's easy to see that for any species tree, any gene tree topology can arise, but with a different probability. It's, I said it's all stochastic. And the gene tree probability is a probability of observing a binary gene tree topology, TG. TG is a gene tree topology under a given species tree with a branch length, TS. And intuitively, the larger this gene tree probability is, and you are more likely to observe this gene tree topology TG. And it has multiple applications in inference in both population genetics and also phylogenetics. So for computing this gene tree probability, if the trees are small, people actually have done calculation by hand, and this is some classic papers and trace back to more than 30 years. And they usually just consider drawing on a paper and say what kind of scenario can happen. And but for larger trees, and the algorithm is neat. And it's because nowadays we have hundreds of, maybe even thousands of populations or alleles. So in this case, you do need an algorithm. The first algorithm is done by Dagan and Sauter in 2005. It's more than 10 years. And their approach used something called a coalescent history. So coalescent history means that you need to consider, you need to specify for every coalescent event where they occur on the species tree. You don't have to exact pinpoint, but you just need to tell roughly along which species tree branch this coalescent occurred. So, for example, this one. So let's say we have this particular genealogy, and then this means that the first coalescent occurred within time of t, and the second coalescent occurred above t. So this is one particular history. And but it's easy to see that for the same for this particular coalescent, it doesn't have to be occur within time of t. So in fact, if you see this one, this is a same gene tree topology, but it's a different history. So you can continue to trace back. Now, can say that lineage A and B, they do not coalesce, and they actually coalesce here. So then they finally coalesce. So the both coalescent occur above time of t. So this is a given different history. And the approach by Dagan and Sauter is that they simply just enumerate all the possible coalescent history, and which is called H. And then they use this it's a law of total probability, and then you can use a very simple formula. You can compute the gene tree probability, and the reason why they have to consider the coalescent history is due to the following main result. One of the main result, classic result in coalescent theory, and the thing is, if you specify, if you're given a particular coalescent history, you actually fixed for every species tree branch how many lineages on the bottom of this branch, and how many lineages on the top of the branch. So this is this probability of you have say you observe lineages on the bottom of a branch, and then you observe after some coalescence, and then now you'll see there's only like a V lineages left after time of t. This actually has a nice closed form formula, and this is done by also more than 30 years ago, and it has a closed form formula. And for anyone who has know some coalescent theory, you'll probably see lots of formula. But I think this is the one. Is one of the one I found is one of the most useful one, at least for my research. And so. essentially, once you fixed a coalescent history, then the probability of this gene tree topology is essentially a product of this PUVT. Again, PUVT is the probability of U unlabeled lineages coalesce into unlabeled lineages within time of T. So let me give an example. And so recall this is a PUVT. U lineages coalesce into V lineages within time of T. And this is a particular species tree, and now we have this gene genealogy with a fixed coalescent history. And now you can see, let's see how we can compute the probability for this particular history. And I said for this particular history, the probability is equal to P21 of T1. Why P21? You have two lineages below, and then you have one lineage on the top. So it's P21 of T1. And then you consider another branch, this branch of time of T2. And you have two lineages on the bottom. You have two lineages on the top. So it's P22 of T2. And then you time of another factor, which is on the very top. Now you have three lineages, and you have one lineage on the very top. So you have infinite time to coalesce. And this is pretty much all. And only little thing is there are some other technical details called some kind of a combinatorial factor I didn't really tell you. And the main challenge here, however, is that the algorithm for Declan-Salter is that you have to enumerate all the possible coalescent history. And I took a snapshot from the paper, and where they show how many histories there will be for some simple topology. And you can see here, for 20 population or 20 texts, there is 1.7 billion coalescent history there. And you can see that the numbers grow very fast. So it's, on this, a couple years ago, I also developed another algorithm called Stealth algorithm, and which is it's based on something in computer science called dynamic programming, and it's usually it's much faster than the Declan-Salter's algorithm, but it's still exponential. So on this point of time, and there's no known algorithm that runs in polynomial time. We can compute the gene tree probability very fast. I still do not know how to solve the general case, but I recently found one way to, for like a restricted case, I can have a polynomial time algorithm for computing the gene tree probability. And this restricted version is motivated by the population genetics, where you have many gene alleles, but you only have a small number of populations. So in 1,000 gene project, for example, you have like 26 populations, but there's haplotypes from over 2,000 deployed individuals. So that means usually people sample a lot of alleles from a population. And the question is, can we compute the gene tree probability efficiently when there's a large number of gene alleles, but the number of populations is small? And just like showing up here, this is showed like two populations. And in this paper, I show that this is if we consider the number of populations is fixed to a constant, then the gene tree probability can be computed in polynomial time, exact. The basic idea is we can merge the coalescent history, multiple coalescent history into one, which I call the compact coalescent history. So as shown up here, and let me just give you a very rough idea what is a compact coalescent history. So this is based on the so-called upper lineage count. And this is basically the number of lineages you observe on the top of a on a species tree or population tree branch. And let's consider this coalescent history. And here, I say ULC, upper lineage count. In this case, ULC equal to 3. You have 3 lineages on the top of this branch. You have 1 lineage on top of this one. You have 1 lineage on top of this one. So combine this, what is a compact coalescent history? Compact coalescent history essentially is the ordered list of this upper lineage counts for each population tree branch. So it's simply just 311. That's why I call this CCH, compact coalescent history. And the important thing here is that multiple coalescent history, you can actually obtain the same compact coalescent history. So in this case, this is a different. This is a different coalescent history because you can see the order of coalescent occurred. D and C are different. So, but they have the same compact coalescent history. This is like upper limit counts to 3, 1, and 1. So they get to the same CCH. And I show that in my paper that you can, the number of the CCH is really much smaller, and it's actually, and also you can compute gene tree probability purely based on the CCH, not the previous coalescent history. And just quickly convince you, it's really polynomial bounded if the population number is a constant. And so remember that CCH is just an upper limit count. So we have, in this case, we have this CCH, C1, C2, C3, C4, C5, and listening to this CCH. And note that each one of them is no bigger than n. N is the number of gene lineages. So when the number of populations is a constant, then the length of the CCH is constant. So it's a polynomial bounded. And this figure shows that the algorithm really runs scale much better than the original stealth algorithm, which is currently the best algorithm for computing the gene tree probability. And you can see as the number of the horizontal line is the number of alleles per population. I fixed this number of population to be 2, and you can see the new algorithm just scale much better than the original stealth algorithm as when the number of alleles increase. The last one is quickly show you what we can do with the new algorithm. And I can show you how we can use the new algorithm to infer the so-called population tree or the population divergence history. And here I can go with population haplotype to infer the gene genealogy. This is based on the assumption that there's an infinite size model and also there's no intra-locus recombination. And so here is something called perfect phylogeny, and I don't have time to explain that. And once you obtain this gene genealogy, and you can then use the new algorithm to estimate how for this pair of population A and B when they diverge. So this is used like a maximum likelihood estimate based on the computed gene tree probability. And so you infer the divergence time, and then this will give you like a pairwise population distance of all the populations. And once you have a pairwise population distance, you can use say standard method like a neighbor joining to construct the population tree. And in my paper, I show that this actually perform better than the method published recently called Trimix, and which doesn't use this gene tree, which I believe has important information about the linkage disequilibrium. So I guess that's all my time, and so I just stop here. 1.5 minutes. Do you have any more? I guess that's all. No, that's all.